In laboratory today, we're going to continue with our jellyfish green fluorescent protein gene cloning experiment. I would like you to bring your DNA technology handout again. I'm going to do part of that as the pre-lab lecture. And also during today's laboratory period, I'll give you your exams back. It was kind of a mixed bag. I think for a few people it was one of the harder exams. A few people got their high score, but a few people also got their low score, at least near their low score, and I'll give you to look at in today's lab. For today's lab period, I want to start by reviewing the population genetics stuff we went over last time. And remember, the population genetics topic is going to be on the final exam. And then after I review some of the things I presented last time, I'm going to have you guys do some worksheets on it. These are just like the ones that we did in laboratory for the genetics labs. There are some problems here. Please don't actually write on these sheets. Write your answers down on a scratch paper, and then come on up to the front desk here if you want to know the answers, and I'll have an answer sheet. But all right, let's start by reviewing some of the concepts from last time. This idea of population genetics is studying genes and alleles, just like we did when we went over the genetics chapter, but it's more looking at what alleles and genotypes are in entire populations instead of just individuals. The populations and include organisms that are in one area and mate with each other. And one of the concepts that came up last time was something called allele frequencies. And I think I gave you some genotypes from a population that I just made up. It's supposed to be elephant seals, but it was entirely fictitious. I said within this population there were 6 individuals who are homozygous dominant, 38 individuals who are heterozygous, and 56 individuals who are homozygous recessive. If I did that math correctly, it adds up to 100 individuals. But remember, since each individual owns 2 of each gene, everybody has 2 of each gene, there's actually a total of 200 alleles. The number of alleles is always twice the number of individuals. And so remembering that we have 200 alleles, the allele frequencies are what portion or what percent of those total number of alleles are each of the alleles, the dominant and recessive. And when we did the math on this, boy, what did it work out to be? Oh, here it is. For the dominant allele, it turned out to be 25%. And you might remember, I advised you for these types of problems to write the percents in the decimal form, like that. And the frequency of the little allele within those 200 alleles was 75%. And then over here, I talked about something slightly differently. I talked about things called genotype frequencies. And genotype frequencies are merely saying what percentage of the entire population is any particular genotype. And of course there are 3 genotypes: homozygous dominant, heterozygous, and homozygous recessive. And so these were pretty straightforward. There's 100 individuals, and so since 6 of those 100 are homozygous dominant, we could say the homozygous dominant frequency is 6%. 38 of those individuals are heterozygous. So that turns out to be 38%, 0.38. And 56 of those individuals are homozygous recessive. So their frequency is 56%. like that. Some interesting facts about the allele frequencies and the genotype frequencies. If these two alleles, if neither one helps you survive better than the other, if neither one harms you or helps you more than the other one, and if all the mating in the population is random, nobody's choosing their mates based on big allele or little dominant or recessive allele, then the allele frequencies will stay the same from one generation to the next. The dominant alleles don't somehow drive the recessive alleles out of the population. Whatever the population has for the dominant and recessive allele frequencies will stay the same as long as these conditions are being met right here. And another thing about this is that the frequency of the two alleles added together always has to equal 1. You can see that right there. That's just sort of basic logic, I guess you'd say. Since those are the only two choices, the grand total of all the choices has to always equal up to 1. And going over here, these are the genotype frequencies. If a population has been breeding with itself for a long time, for several generations, for any given allele frequencies, eventually the population will settle down into certain percentages of genotype frequencies. And how many generations does it take for a population to come to equilibrium, so to speak, to settle down into certain genotype frequencies? Well, I don't know exactly. I guess there's a certain amount of randomness, but certainly within 3 or 4 or 5 generations for a given allele frequencies, it should settle down into certain genotype frequencies. And there's an equation that allows you to relate the genotype frequencies to the allele frequencies. And the equation is sometimes called the Hardy-Weinberg equilibrium equation. And this is the way it's normally shown in textbooks, like that. And I think that's the way I put it in your notes, but it always seems to confuse students when I show it like that, and so I tried to show it slightly different this year. What it's really saying is that if you take the allele frequency of the dominant allele and square that number, that equals the allele frequency of the homozygous dominant genotype. And if you take 2 times the dominant allele frequency times the recessive allele frequency, that equals the heterozygous genotype frequency. And if you take the allele frequency of the recessive allele and square it, that gives you the genotype frequency of homozygous recessives. Like that. And if you add these 3 together, it's going to equal 1. And the principle there is, again, that if you have all your possibilities, everybody's either homozygous dominant or heterozygous or homozygous recessive, those are your only options. If you add all the possibilities together, they always add up to 1. And you can see from the percentages that would be true. So this is called the Hardy-Weinberg equilibrium equation. And usually it's presented like this, where P equals the dominant allele frequency, Q equals the recessive allele frequency. But as I said, I think it's a little bit confusing, so maybe think of it like this. All right, so let me show you a problem that makes use of this Hardy-Weinberg equilibrium equation. Earlier in the semester, I told you about a genetic disease called PKU, phenylketonuria. And remember this one, it was a disease usually diagnosed in newborn babies, where the babies can be harmed by a certain amino acid called phenylalanine. And if the baby has this disease, where they can be harmed by this certain amino acid, we said that the baby had this for its genotype. There's an enzyme that helps break down this phenylalanine amino acid, and most of us have that enzyme. Most of us are either big Q, or big Q, little q. So we're protected from that particular amino acid. But if the baby is born little q, it does not have a functional copy of the enzyme that breaks down the phenylalanine amino acid. So the phenylalanine builds up to too high levels in the baby's brain, and mental retardation can result. And what they do, if a baby does have PKU, they just put the baby on a special diet so that it gets very little of that phenylalanine amino acid. And then at a certain age, I don't know exactly how old, 7 or 8 or 9, the kid can start having phenylalanine again because the brain has developed sufficiently at that point. This is a fairly rare disorder in the population. And as it turns out, about 1 in 10,000 babies, only 1 in 10,000 babies is born with this PKU disease. So, if I tell you that bit of information, which bit of information have I told you? Am I telling you that, or that? Which one of those 5 am I telling you? Homozygous recessive, yes. Anytime there's a recessive genetic disorder and you say this person has the genetic disorder, you know that they're little q for the disease. And so if I tell you that the frequency of that people with PKU is 1 in 10,000, I just told you this bit of information right here. Now, how do we write 1 in 10,000? What is it, 0.01. I think that's what that is in decimal form. So, I just told you that. What then is the allele frequency of the little q allele? It's going to be the square root of the genotype frequency, right? Because that's squared. So who's mathematically minded? What's the square root of 1/10,000th? No, you're going the wrong way. Yeah. All right, because if you square that, if you square 0.01, which is 1/100th, you get 1/10,000th, 10,000ths, like that. All right, so we've just made kind of an interesting observation here. From the frequency of the little q genotype, thanks to this Hardy-Weinberg equation, we were able to figure out the allele frequency of the little q allele. What's the allele frequency of the dominant allele, big Q? Point 0.99. Right? Because we know that the 2 allele frequencies must add up to 1. And so if we know that this was 0.1, this has to be 0.99. What's the allele frequency, or what's the genotype frequency of carriers for this disease in the population? In other words, for people who are big Q, little q. It's going to be 2 times 0.99 times 0.01, which I have no idea what that is in my head. Looks like around 2%. Or I should say 0.02. And so 2% of the population are carriers of this PKU. And if I say, well, what portion of the population is homozygous dominant? In other words, what portion of the population is big Q for this disease? What's that? Well, we know that the frequency of the dominant allele, oh, I should have put big Q there. The frequency of the dominant allele is 0.99. And so I square that, and it looks like 98%. And within rounding error, that does add up to 100% like we'd expect. So let me do a quick recap of that. If someone tells you what the frequency of people who have a genetic disease, a recessive genetic disease is, they're actually telling you what part of the population, the genotype frequency of little q, or homozygous recessive. And once you have that, the allele frequency of homozygous recessive, you can use the Hardy-Weinberg equation to say, aha, once I have that number, if I take the square root of that number, I've got the allele frequency for the recessive allele, 0.01 in this case. Once you have the allele frequencies for one of the alleles, you can automatically find the allele frequency of the other allele.